Dear students, in this module, I'm going to introduce you the topic protein folding. Why is protein folding important? And what are the mechanisms that are involved in the folding of proteins? All of these questions are very important if you are to study any biological process. The reason for that is that all biological processes are determined by the proteins involved within them. Now, the proteins, they function only because they have this folded or 3D structure or a form. Therefore, any anomaly in the folding of a protein will definitely alter the effects of the biological process due to a lag in the protein structure participating in the process. So let's start this module with an introduction to how the protein folding begins. You know that there are 20 different kinds of amino acids, and that these amino acids, they come together as beads and they polymerize to make chains. So once these chains of amino acids or polypeptides are formed, then these chains, they fold upon themselves and take 3D packed structural forms. These structural forms are formed during the process of protein folding. But how does a protein actually fold? It is a question that the scientists have been hunting for a long time, and it still remains unanswered. The reason for that is that there are so many possibilities that are there for a protein to fold that it is simply impossible to actually compute all of them. So, if you cannot compute all the possibilities of how a protein will fold, obviously you cannot select the best one. So this has been quite a dilemma, and lots of people, lots of research groups in the world, they are focusing on this problem. So after the polymerization happens and the amino acids, they come together, there is this linear chain of amino acids. Now, if you were to fold it upon itself, and that, as I just told you, there are so many ways to fold, then how exactly are you going to proceed with the problem? This was met by one of the very famous scientists, Anfinsen, and he also struggled with the question. And even more painfully and interestingly at the same time, he observed that the proteins, upon being mixed in a solution, they folded into their native forms very quickly, as if the proteins knew how they were to be folded. So this was very perplexing and very confusing for Anfinsen as well. This was especially true after he realized that the protein that is in its folded form is in the minimum energy or minimum free energy state. How do the proteins compute what is the overall energy of the molecule, of the folded structure? And then how do they search for better structures which have a lower free energy? So these are all very interesting questions which are still unanswered till date. In this diagram, I'm going to elaborate on Anfinsen's dogma, which essentially states that the protein is going to fold to a state where it has the minimum free energy. If you look closely at this landscape, then this is the amount of free energy. So free energy is small here and large here. So as the protein enters this landscape, it is continuously decreasing its free energy. So as it keeps moving and enter into the valleys, then it reduces its energy further. Now, if a protein enters into a local minima as highlighted in blue, then it has to jump over this local maxima in order to continue its journey from the large to smaller free energy state. Of course, eventually it is going to arrive at this state, and here you will have the minimum free energy for the protein structure. So this was Anfinsen's dogma, but then again, if this were to happen, how do the proteins achieve such a computational power, or how do they arrive at the energy levels that are the minimum for a structure? And secondly, how do they fold to arrive and achieve that minimum free energy? So all of these questions are very interesting and very pertinent to the protein folding, and the take-home message would be that the proteins, they fold spontaneously, they fold very quickly, and they do not evaluate all the possibilities for structural conformations. So therefore, one can safely assume that all the information required for folding a protein is actually there within the amino acid sequence. And therefore, the proteins, they achieve thermodynamic stability because they have the lowest free energy. And lastly, the proteins fold to organize themselves to perform the various functions that they have to undertake within the cellular life.